No, es largo.
Está bien, está bien.
¿Alguna vez te ha pasado que alguien te da una larguísima?
Es la primera vez que presento a alguien.
Ah, vale.
Por-
Bien.
All right, I think we can start. So for today's seminar, we have Pablo Finkel. Pablo obtained his undergraduate degree in immunology in the University of Glasgow, Scotland, where he focused his work on the interface between the nervous system and the immune system. Then he proceeded to his master's degree in immunology and microbiology at the University of British Columbia in Vancouver, Canada, in the hope of learning microbiology to eventually make the jump to astrobiology. And now he's finally pursuing the PhD he wants on a topic he likes in a place he enjoys. And today he's going to talk about lipid biomarkers in extreme environments with the relevance for mass explorations.
Thank you, Laura. Can you hear me well? More or less? You can hear me, right? Okay. Okay, so thanks, Laura, for... It's green. Maybe we can put it a bit louder. Probando, probando. Whoa. ¿Y ahora? Okay, thank you. Okay, so thanks, Laura, for the introduction. So today I will tell you a little bit about lipids that we look for in places on Earth that have extreme conditions for most of life as we know it. And we are going to extrapolate all this information to Mars, to the search for life on Mars. This presentation here is based on a review that we have written in 2023. It's this one. It's in my desk. It's printed. If anyone wants a copy, it's in my drawer. It's been there for 2 years. So without further ado, the way we're going to start this presentation is with a long and thorough introduction, so everyone can understand the organic chemistry of lipids a little bit. We're going to jump through different extreme environments, hot springs, hypersaline environments, and poly-extreme environments, and we're going to do a quick summary at the end. So the reason why we know a lot about Mars now is because the search for life on Mars has been an evolving idea. We started with the Viking program. Now, I'm not going to put every single mission that has gone to Mars here, but it's going to give us a little overview of what has happened. So the Viking program was the first mission to land on Mars, and the actual goal was to find living organisms, or part of the goals. And not only didn't find life, also it got an interesting false positive. So what scientists thought is that maybe we went too far too quickly, and we need to learn a little bit more about the red planet before actually trying to find evidence of life. That's why the next missions were changed a little bit in their goal. The Mars Pathfinder and the Sojourner rover. We have a model of this in the entrance of CAB. They had some experiments, but they were mostly proof of concept missions where we wanted to demonstrate. That one's better, right? Hola, hola. Here. So is that good? A little bit lower. Probando. Yeah, there we go. So far away from me. So these missions were trying to demonstrate that we can go to Mars and we can move around a little bit with a rover. Then came the Spirit and Opportunity Mars exploration rovers. They were very interesting missions that did a thorough geochemical examination of the surface. They went to very interesting sites on Mars, very chemically diverse sites. Then the InSight lander did a very thorough geological survey of the interior dynamics of Mars until we reached the Curiosity and the Perseverance rovers, which are, after learning all of this, now we can take the search for life into a more meticulous mission of finding molecular evidence of a past extinct life. So that's why we have learned a lot about Mars. There's a good scientific consensus that at this point that the crust might have been habitable billions of years ago. Closer? There we go. Where we envision a cold and wet Mars that has progressively been cooling until the hyper-arid, really cold Mars we know today, the Amazonian Mars, where the conditions are presumably uninhabitable. If we have to envision a point where life might have developed on Mars, it's definitely at some point here. And if we have to imagine a Martian life, well, it's hard to imagine life as we don't know it. It's just conceptually easier to imagine life as we know it on Earth, especially primitive life on Earth. So if we have to imagine some hypothetical Martian bacteria, we can perhaps think about similar biomolecules that are inside this putative Martian life. This could include nucleic acids, amino acids that make up proteins, or a layer that separates the interior of this Martian organism from the outside. So this would be a membrane in the case of Earth. And on Earth, these biomarkers compare between themselves interestingly, because while DNA is very taxonomically specific, meaning if you find DNA on Mars, you're going to be able to pinpoint the biological source easily, it's actually quite labile, which means it's easy to degrade. Whereas lipids, membrane lipids, they are not that specific, but they are very, very resistant. So if this was the period where there was water on Mars, this is probably the period where there was a hypothetical life existing. Genetic material obviously coexisted with this hypothetical life. Maybe it lasted a little bit longer. Amino acids are expected to be more resistant than nucleic acids. And then this is the reason why in our group we focus on lipids in astrobiology, because membrane lipids are expected to be amongst the most resistant biomarkers. Defining a lipid is actually quite complicated. Normally in biochemistry, we define molecules in terms of function and structure. And you'll see that with lipids, it's a little bit ambiguous. Let's start with function. The problem with lipids is that they do a lot of different things. So for example, the hormones that we secrete in our organisms, those are lipids. So the hormonal system is based entirely, almost entirely on lipids. The fat tissue that we have, the fat tissue that keep penguins warm in Antarctica, is made out of cells that have lipids inside. So this is a really important role of lipids, which is insulation in higher organisms. And then the formation of the biological membrane. And this is another reason why lipids are good in astrobiology, because every single cell on planet Earth that you can think of has a lipid membrane. These lipids are different, but they have a lipid membrane. So we cannot really define lipids by function if we want a nice dictionary definition. But by structure, however, we see a similar problem. Lipids are incredibly diverse. They can be simple, they can be really, really complex. But I'm going to ask a question to the audience now. It's not a chemical question, just in terms of what you see. What similarities do you see between these molecules? There's something that they have in common. Anyone would like to guess? Yes? They are long, but what if I make a short one? Like this? It can be short. That's the point. So if the answer I was looking for was around that, they all have this zigzag. And this zigzag is the hydrocarbon chain. And this is what makes lipids indeed hydrophobic. So the reason why oil doesn't mix with water is because of the hydrophobic hydrocarbon chain. So this is something we can use to define lipids, even though they're very different. We can define based on structure and say that they are a diverse group of organic compounds that are grouped together because they do not interact appreciably with water. And appreciably is important, the word, because some lipids do interact with water a little bit, like in the membrane. This is a biological membrane. The hydrophobic part that you mentioned is inside the membrane, and the polar part or the hydrophilic part is outside the membrane in permanent contact with water. So that's why they group themselves like that. So let's delve a little bit more into the biochemistry of the membrane. Here we have a cell, and we have a cross-section of this membrane, and we see all of these guys. And if we zoom on one of them, this is a really big molecule. And this big molecule has a phosphate group. And this is a shout out to astrochemists. Phosphorus is really important in biology. This is one of the three major reasons. It's a really important part of the membrane. So there's the importance of phosphorus. These are fatty acids that I'm going to talk about right now. This is the hydrophobic part, and an alcohol that links them, forming a molecule called the phospholipid. So all of these are phospholipids. And the phospholipids, they have a hydrophilic group and a hydrophobic group, and that's why they arrange themselves in two layers, forming a lipid bilayer that forms every single membrane. Molecules that have a polar and an apolar group at the same time are called amphiphilic or amphipathic, but amphipathic sounds worse, so I say amphiphilic. The lipids that we're going to talk about in this presentation, however, are simple. They're not going to be that complex, except one or two. And we're going to start because we're looking for them on Mars, right? So we cannot really look for something that is really complex. The simplest ones are the normal alkanes. The normal is, the N stands for normal. And alkanes are just linear hydrophobic hydrocarbon chains and hydrophobic. And we, in organic chemistry, express them with these zigzags. They can be short, they can be long, they can be longer. This one has 17 carbons. We can decorate them with methyl groups. This would be a dimethyl alkane because of the two methyl groups. So these are quite simple. Then we have the fatty acids. The fatty acids are also quite simple. They consist of a hydrocarbon chain, but if we add a carboxyl group, then it's an acid. So this is a simple thing, a nice, beautiful thing of organic chemistry. Whatever you have, you put a carboxyl group, it's an acid. You remove it, it's not an acid anymore. And here we have a saturated fatty acid. And here we have an unsaturated fatty acid because it has an unsaturation, which is also a carbon double bond. So when you add these unsaturations, sometimes you can introduce bends in the molecule. And this is like this one over here. These kinks or these bends are really important to adapt to extreme conditions. You'll see why. So if we go back to the phospholipid, now we know that the two chains over here are indeed saturated fatty acids. And fatty acids, one is saturated, and the other one has a double bond, it's spent, it's unsaturated. And same deal with alcohols. Alcohols, whatever molecule you have, you add a hydroxyl group, you can call it an alcohol. If you remove it, it's no longer an alcohol. And I've just included 3 different molecules that one or two of them at least might ring a bell. And that's because they just have this hydroxyl group. Now, alcohols can get really complex. So for example, this one over here is called caldarcyl, and it has 2 hydroxyl groups. And this molecule is actually really important for the membrane of archaea, which I would also talk about in this review. Right, so to sum up the introduction, something that I really have to mention here is the concept of degradation. Now, these points in time are problematic because, yes, lipids are resistant, but like every other biomarker, they degrade over time. So there's a point where they disappear and we can no longer detect them. And what would happen with a membrane, for example, is that we have here the membrane. The membrane degrades into individual phospholipids that eventually break down into their fatty acids. These fatty acids can even lose the carboxyl group, become linear hydrocarbons that even break further into smaller fragments. And these fragments, which are expected to be found on Mars, and they have been found on Mars already, these simple organics are too complicated to associate to life, and these are the factors that can cause this. So heat, pressure on Mars, definitely radiation, oxidants like the perchlorates on the surface of Mars, water, microbes, many things can destroy our molecules of interest. So we need them to be preserved. If we want them to reach the point where we can look for them, we need to look for them in good matrices for preservation, like salts, ices, and minerals like phyllosilicates. These are phyllosilicates over here, which are part of the target of the Rosalind Franklin rover for the ExoMars mission, hopefully in 2028. On Earth, this is the classic slide where I say that we cannot go to Mars, the rovers can, and we have to go to terrestrial analogs of Mars. And this is also the classic comment where I say that there's no perfect analog of Mars. There's nowhere on Earth that is remotely similar to Mars, but we can get so close, and we can get important information from locations on Earth that resemble current Mars. These locations have to be very dry, and they have to be subjected to radiation, and also they have to be cold, or in Atacama also can be hot, but definitely cold temperatures help. But we can also go to places on Earth that resemble Mars billions of years ago. For example, some hydrothermal fields on Earth, or even Rio Tinto, because Rio Tinto here in Spain has a really, really interesting chemistry that I will also talk about. You might be wondering, wow, he says he's going to talk about so many things. There's no time. I fear time. So I'm going to try to go efficiently. Now, this is a very important slide, one of the most important slides. There are no specific lipids for specific extreme conditions. That's something I'm telling you from the get-go. Lipids that we can find in hot conditions can be found in hypersaline conditions, but there are patterns, and there are patterns that can be somewhat exclusive to one condition or the other. What there is specific ways of preserving lipids in specific extreme conditions, and that's one of the important points of this talk. So without further ado, I'm going to jump to the first chapter, which is hot spring environments. And hot spring environments are extremely relevant for astrobiology for a number of reasons. First, hydrothermal activity is inextricably linked to planet formation and evolution. Most of the bodies in the solar system have or had hydrothermal activity, or at least geothermal activity. These are interesting hotspots for the emergence of life. It is a debate whether this happened in the bottom of the ocean or in a little pond in the surface, but definitely hydrothermal activity is up there. And also, these locations on Earth, they have unprecedented mineralization rates, which means that they keep precipitating minerals that have the capacity to preserve these organics really well. This is a really nice sign of some of the hydrothermal features in Yellowstone. And I like it because it explains them quite well. And we're going to talk about geysers mostly. And this is how they work, right? They normally have an underground pool that accumulates with really hot water. And this pool has a constricted path. So when this fills up, the heat and the pressure accelerates the water through this constricted path. And that's why periodically the geysers expel water. We're going to focus on geysers that are made out of or expel silica sinter, which is a type of mineral. But there's also a lot of chemical variety in geysers. They can be made of carbonates, or even they can be iron rich. So here we're going to talk about the membranes. We're going to go back to the unsaturations that I told you before of the fatty acids. Now, if you need organisms to survive at really high temperatures, you normally want a rigid membrane. And a rigid membrane, you can obtain it if you don't put a lot of double bonds, if you don't put a lot of methyl groups, if you keep a nice, long, saturated chain that is quite rigid, that can resist heat better. However, if we talk about really cold temperatures where psychrophiles can exist, if we introduce double bonds, if we introduce methyl groups, the membrane is going to be more flexible, and that's better to withstand really cold temperatures. So what are we looking for in organisms in thermophiles? We're looking for long lipids that are not very saturated. So here I've drawn a geyser. This is the best I could do. And we're going to talk about 3 different temperature areas. So near the vent, we have the highest temperatures. The distal aprons closer to the vent have also hot temperatures, and then we have cooler geothermally influenced terrain. So near the vent, we're going to have thermophiles. Further away, we're going to have thermotolerants, which are organisms that are okay with heat. They'll make it. And we have pesophiles that are a larger group of organisms that live at broader temperatures. So the thermophiles, I told you before, they need long saturated chains. So here we have a long decarboxylic acid, and it's an acid, remember, because of the carboxyl group. This one is called diabolic acid, and it's just a funny name. The diabolic thing has nothing to do with the fact that it belongs to thermophiles. It's a chemical name. And also long chain isoprenoids, again, a long chain that doesn't have a lot of double bonds. The thermotolerants are going to have also lipids that are nice and long, like the beta-carotene or long alkanes, but maybe with a few more unsaturations. And in this case, way more. I have put beta-carotene because the example I've chosen, the chloroflexi, can look orange. That's why they have beta-carotene. And then the mesophiles, like cyanobacteria, are going to have lipids that are found in most cells. So the dimethyl alkene I showed you before, a monounsaturated fatty acid, and this crazy alcohol that is a rigidifying agent in the membranes of many bacteria. This is great. We now know a lot of lipids that we might be looking for, but how do we preserve them? Well, if we go back to the mineralization of geysers and we focus on silica sinter, we see that this mineral, opal A, which is basically a hydrated and amorphous form of silica, is excellent at preserving organics. The problem is that this mineral can crystallize into opal CT and at more advanced forms of crystallized opal and eventually develop into quartz. And once this mineral crystallizes into quartz, the organics that are preserved inside are absolutely obliterated. So crystallization is our enemy, and amorphous hydrated minerals are our friend in terms of preservation of organics. On Mars, silica sinter is a thing. It exists. There's a big crater called the Gusev crater that was explored by the Spirit rover. And inside the crater, there's this feature called the home plate. Inside the home plate, they found a very interesting formation of silica sinter. Now, I'm comparing in these images, home plate is on Mars, and El Tatio is a hydrothermal field in Chile. And I want you to focus on the last photos here. This is Mars, Chile, Mars, Chile. And look at the formation here. This is probably amorphous silica sinter that looks very similar. From here, we can conclude that there are silica deposits on Mars. And the lipid biosignatures that have been detected at Chile, at El Tatio in Chile, they might have been similarly entombed on Mars when these silica supersaturated fluids came out and rapidly cooled down, precipitating into the minerals. So this is an interesting mission target. In fact, if I'm not mistaken, there is interest in returning to the silica deposits of Gusev crater, also because the Spirit rover didn't last as much as they wanted it to last. So in conclusion, what can we look for in these environments on Mars? Definitely long saturated lipids, but these features, the carboxyl group, are probably going to be degraded over time. And these branches in the molecule I showed you before are also probably going to be lost over time. So keeping the stuff realistic, probably looking for long saturated carbon chains is already a very interesting target. Length is also interesting in astrobiology because carbon chains that can form abiotically in the interstellar medium, for example, they tend to be shorter than those that biology can form. Right, so that's the little conclusion that we draw from hydrothermal environments. Now we're going to move on to hypersaline environments. And we're going to focus on the Atacama Desert. Normally people talk about the hyperarid conditions of the Atacama. Here we're going to focus on the hypersaline conditions. And we can find salt units in the surface of Atacama in numerous salt flats, but also there are halite units that are underground. So in this image, you can see from this paper that these vertical lines represent halite units. Sulfates are also salts, and they are also found on Mars. Now, there's this type of organism called archaea, in this case, halo archaea, because they like salts, that really tolerate and like high salinity. And there's a lot of evidence, this is really well established. There's a lot of work on archaea that thrive in hypersaline conditions, specifically in the Atacama Desert. So let's talk a little bit about archaea. I get really excited with archaea. Here we have the tree of life, and we have LUCA, the last universal common ancestor. And we have three main domains in life. We have bacteria, we have archaea, and we have eukarya. We are made of eukaryotic cells. These are the most complex and evolved cells. Now, if you look under a microscope to bacteria and archaea, you're no chance you're going to distinguish which one is which. They're actually very similar. Genetically, though, they have been grouped in a completely different place. So they are not at all similar. Also, another difference between bacteria and archaea is the membrane. Seriously, the membrane of archaea is insane. Super different to the membranes of bacteria. And the membranes, the lipids in the membrane of archaea are based on this unit, this 5-carbon unit called the isoprene. The isoprene always has a branch in carbon 2. If we put together a lot of isoprene units, we're going to form molecules that look like this. These are called isoprenoids, and they have consistent methyl branching along the chains. This is a key feature of archaeal membranes. So if we go back to the phospholipid molecule I showed you before, this is in bacteria. The bond, the chemical bond that connects the fatty acids with the polar group is called an ester bond. And this is how they group together, forming a bilayer. However, in archaea, we have this crazy molecule that I showed you before. It's this one. It's just a different configuration. Remember the caldarchial. Instead of ester bonds, it connects the lipids to the phosphate group with an ether bond. And ether bonds are way more resistant than ester bonds. So that's good for extreme conditions. And this is how the molecule looks like. You'll see that it's one single molecule instead of two. So archaea, extreme archaea, don't have a bilayer. They actually have a monolayer in their membranes, right? So if we look at this photo, I like this diagram a lot. Not a photo, sorry, diagram. You can see that an archaea have a lot of these molecules in the membrane that span the whole length of the membrane. And here I'm just showing it differently. So the more extreme the conditions, the more long, where you are, there you are. The more long isoprenoids forming a monolayer you're going to have. So that's why the archaeal membrane looks like that. It's so different to the bacterial membrane that sometimes I wonder whether there was a last universal common antecessor for bacteria and another one from archaea. That's something we don't know, but it's definitely super interesting. But it's beyond the scope of this talk. Now, here you have some plots, the only plots I'm going to include in this talk, where you see that all these molecules, pristane, phytane, crossetane, squalane, these are isoprenoids. They can be found in archaea, but they can also be found in bacteria. But you can see that they're concentrated in the blue area, which is the halite unit. So where the salts are, the isoprenoids can be found. And here is a work from some scientists from NASA. And this is a subsurface halite unit in the Atacama Desert. And you can see that the isoprenoids peak specifically in these halite units, right? So normally you would expect to find archaea there. That's why there's a higher concentration of isoprenoids. Now, this is very relevant to Mars. There are evaporitic deposits on Gale crater. This has been detected by the Curiosity rover. There are carbonates, there are lots of sulfates there. And these evaporitic deposits, these brines have formed because there have been very rapid evaporation rates at some point. Because the Gale crater in the past was a lake, Gale Lake. Just like Jezero crater, it's a paleo lake because in the past there is solid evidence for water activity. So maybe there were archaea-like organisms that they may have left some sort of biosignatures that have been able to be encapsulated in the salts of Gale crater. So what we could try to look for on Mars, don't worry, it's not that, because these phosphate groups are definitely going to be lost quickly. This kind of degradation can be observed on Earth, so imagine on Mars. But even the resistant, where are you? The resistant ether bonds that keep together the molecule are going to break because of the billion-year timescale and the radiation and the oxidants of the surface. So if we are optimistic and at the same time realistic, it would be interesting to look for short chain isoprenoids on Mars. So yes, maybe the methyl branches, these branches here. Yes, they definitely can be lost, some of them. But if we find on Mars at some point a hydrocarbon with some branching, with some methyl branching, these kind of configurations are abiotically unlikely. Possible, but unlikely. Abiotic mechanisms normally prefer to add carbons one by one to the end of the chain. So methyl groups are also our friend. Now, with this little conclusion, we have from the previous chapter, long chain hydrocarbons, good target. Short chain hydrocarbons with some branches, good target. Long chain hydrocarbons with branches, you go nuts, but these are probably more unlikely. And the final objective could be to look for linear isoprenoids. Now, if we move on to the third and last chapter, I'm going to talk about polyextreme environments and what we can look for in environments such as this. And I'm going to talk about two because of time issues. One is Rio Tinto, Spain. So I had the chance to go with some people from CAB a year and a half ago or something like that, and I loved it. So if you haven't been to Rio Tinto, it's beautiful. And the other one I really want to go. It's probably the most extreme location on planet Earth, which is the Dalol hydrothermal system in Ethiopia. And you can call it a hydrothermal system, but you can call it a desert. You can call it a hypersaline area. You can call it whatever you want. You can call it a super acid, horrible place. The conditions in Rio Tinto are quite extreme indeed. They are sulfur-rich. They have sulfur-rich mineralogies. The pH of the river in average is quite low. And there's a high concentration of heavy metals. But if we go back to Ethiopia, it's also sulfur-rich. The pH can be negative. This is something I learned a few years ago. pH values can actually be negative. Imagine how acidic that is. There is hydrothermal activity. So there's extremely hot water being emitted consistently. It's halide supersaturated. Everything you see here is salt. And the worst thing about it is that it's hyperarid. It doesn't rain. It has a desert climate. It's actually terrible. But it's terrible for most life, right? Not for some organisms that can make it over there. So examples of these organisms are bacteria that can undergo sulfur metabolism. So one example that could possibly be detected in both is this one. This, the sulfovibrio species, is a sulfate reducer. So it reduces sulfate to hydrogen sulfide. Then we have purple sulfur bacteria, for example. These ones do the opposite. They are sulfur oxidizers. So they do the opposite metabolism and they create sulfates. And the beautiful thing about purple sulfur bacteria is that they do photosynthesis. It's actually quite nice. Not only they metabolize sulfur compounds, but they also need sunlight. And then we have some organisms like these ones, like Thermotoga napolitana, that they undergo sulfur metabolisms, but they are also halophiles. They like to live in extremely saline conditions. So there's not a big pool of organisms that can live in these locations, but they are. What are the lipids that we can look for in places like this, in organisms like this? Well, you would think that they are very particular and very unique, but not really. So these acidophiles or sulfur bacteria, they have quite specific lipids, but they're not exclusive to them. They can be found in many more cells. For example, and I'm going to talk only about fatty acids now, this one here, a fatty acids of 16 carbons with a methyl group exactly in carbon number 10 can be considered a biomarker for sulfate reducing bacteria. And also these fatty acids here that have a methyl group in the second to last and third to last positions of the chain. We call them iso and ante-iso. Also, these fatty acids that have a cyclopropyl structure here, these are common lipids in the membranes of purple sulfur bacteria, for example. You wouldn't find this fatty acid everywhere. And unsaturated fatty acids like this one, they are super ubiquitous. They can be found in sulfate reducers, but they can also be found in sulfur oxidizers, cyanobacteria, and many, many more organisms. So let's talk a little bit about where these fatty acids can be preserved. I told you that the Dalol hydrothermal system is terrible for life, but it's actually not, because it has so many bad things that all of these bad things are actually contributing to preservation. It has salts. Salts can encapsulate organic matter. It has silica sinter, which can encapsulate organic matter. What about Rio Tinto? Rio Tinto has this great mineral called ferrihydrite that is also great at preserving organic matter. Now, ferrihydrite is a little bit like silica sinter. I told you that silica sinter was amorphous and hydrated. Ferrihydrite is like the iron silica sinter. It's amorphous, it's hydrated, but it has iron. And there are advantages of ferrihydrite over silica sinter. But to talk about these advantages, we need to make two assumptions. First, assumption number one is that Mars had water. We're okay here. So, good consensus that Mars had liquid water in the surface because you need it to form ferrihydrite. And also, we need to assume that Mars was rich in ferrous iron, which is oxidized with 2 positive charges. Now, most of iron on Mars is ferric. It's iron 3+, but this is iron that has been oxidized from this one. So if, in fact, there is evidence for minerals containing ferrous iron on Mars, but also the fact that Mars has a lot of ferric iron means that, and a hydrated past, means that there is a good chance that these two conditions can be met. So let's assume that these things happen. They're quite plausible. There's good scientific evidence for both. The advantages of ferrihydrite is that when this mineral, when fluids that are saturated in iron precipitate into this mineral, ferrihydrite is actually has a lot of iron 3, ferric iron. So when this oxidation happens from iron 2 to iron 3, this system is going to kidnap any available oxygen in the system to form ferric iron. And this is good because this available oxygen is what oxidizes biomarkers and can destroy them eventually. So maybe it could act against the action of perchlorates. Also, this matrix is positively charged thanks to the iron 3. And the fatty acids of the membrane are actually negatively charged. So they can bind very quickly, and this can really ease the process of mineralization. So you want, normally, there was a good seminar yesterday about it, mentioning that the quicker the entrapment of the organics, the quicker the entombment, the better the chances for preservation. So this helps in that sense. And finally, assuming there is life, or there was life, iron inhibits enzymes that normally destroy lipids. So this is good for preservation even on Earth. Normally, ferrihydrite can crystallize into goethite, which is also amorphous, but it's a more crystallized form of ferrihydrite, and eventually into hematite, which is like the quartz of silica, but it has iron, and it's highly crystallized, and it would obliterate any preserved organics. So again, crystallinity goes against preservation potential. On Mars, there is this place called Meridiani Planum. Meridiani Planum was explored by the Opportunity rover. It was one of the 2 Mars exploration rovers. And you can see in color, these are hematite deposits. So this is the crystallized form of this mineral, our enemy. But if there is a hematite and there was an aqueous past in Meridiani Planum, because this is what the scientific evidence points toward, that Meridiani Planum had an acid and aqueous past, then maybe we can think of amorphous hydrated iron minerals. So there is satellite evidence for iron-rich minerals like hematite, but there is also satellite evidence for iron 3 amorphous components. And it's not clear from orbit if it's ferrihydrite or if it's goethite. But I think there is some work that demonstrates the presence of goethite from the Opportunity rover. So maybe in this environment that has been acidic in the past, and it has had water in the past, maybe there are biomarkers from extinct acidophiles preserved in these amorphous iron matrices. So what can we look for? Now I'm going to be a little bit more optimistic. and a little bit more ambitious. And we're going to talk about fatty acids. Now, fatty acids are elusive molecules on Mars, but they carry a lot of biological information. Now, these fatty acids that I've showed you before, these beautiful structures here are unlikely to form abiotically, but they are also unlikely to be preserved in the billion-year timescale. So these are probably going to be lost. So looking for fatty acids would be probably, it would be one of the most interesting targets, because where are you? The carboxyl group here is also abiotically unlikely, yet possible. But just the presence of this carboxyl group in a molecule carries a good amount of biogenicity. In other words, biological identity. And you might wonder, or you might ask, yes, but aren't these carboxyl groups going to be lost at some point? Yes, probably. It's a weak part of the molecule, leaving just the hydrocarbon chain. But if we look for them in a good preservational matrix, we might find them, because the latest experiments that we've been doing show that under strong radiation, the fatty acids can survive, keeping the carboxyl group. And even in environmental experiments that we have done, where we test different samples that are progressively becoming a rock, progressively fossilizing, one of the most complex lipids that remain is the fatty acid. So in fact, there is even I wouldn't call it evidence, but there is provocative finding by the Curiosity rover that suggests that it could have been a fatty acid, but I saw this a long time ago, and it's not confirmed yet. It actually was shown in a conference. It's not even in a paper yet. All right, so in conclusion, I'm finishing up. If we look toward the Rosalind Franklin mission toward Oxyaplanum on Mars, this mission is going to focus on this mineral here, on phyllosilicates, which are good minerals because they can preserve organics between the layers. So let's keep in mind what we've mentioned today. Looking for long saturated carbon chains would be interesting. We want to go a level higher. We could look for saturated chains with consistent methyl branching. And if we want to go an ambitious level higher, then we can try to see if we can find some fatty acids. I didn't have time in this presentation to talk about one of the most important aspects in the search for life on Mars, which is the classical question. Well, you find it, how do you know it belongs to a living organism? And this is what we call assessing biogenicity. It's key, and it's very complicated. And I don't have time to delve on it, but I will just briefly mention three things that we can do in case we find an interesting molecule such as this one. One is, and this is not something the rovers can do currently, but if Mars sample return works out, and if we bring samples from Mars, we can do compound-specific isotopic analysis to the lipids. Life normally prefers carbon-12. So life really fractionates carbon-12 from carbon-13. Life doesn't like carbon-13. So normally, compounds that are really impoverished in carbon-13 are associated to life. This is very general. There's a lot of extra details. Also, chirality. This is something that the Rosalind Franklin rover can explore. Normally, life prefers molecules that have one configuration over the other one. So we normally call it the left and right configuration. For amino acids, life prefers the left configuration, and for sugars, life prefers the right configuration. If we see that on Mars there is a preference for one configuration over the other one, instead of a mixture of both, then that could be an interesting sign of past life. And finally, if we are assessing a series of lipids, each bar here represents a hydrocarbon chain of increasing carbon length. Normally, if the sample doesn't have life, we're going to find a distribution like this one. But when we find distributions like this, where even carbon chains or odd carbon chains are preferred, that's a sign of biological activity, because the enzymes that build up these hydrocarbons add the carbons in twos. that's how it is. Whereas abiotic synthesis adds carbons one by one. So that being said, assessing biogenicity is fundamental. And with this, I'm going to finish this presentation. So thank you so much for your attention.
Thank you very much.
You can.
Thank you very much, Pablo. Very general seminar. Thank you for non-expert. I think it is very useful, such a kind of seminar. So now it's time for questions. I also have two, but we can start from the audience.
Okay, so it was a very nice talk. Congratulations. Didn't know about the this membrane of the archaea. It was like something I learned today. But I was going to ask you a question about the degradation of these biomarkers, these lipids used. Well, we expect, or that's plausible to find, right? So in the end, we are like looking for them in the surface of the planet, right? Like you mentioned evaporites, you mentioned, the surface in general. And yeah, it makes all the sense in the world that the most interesting features of those molecules that get lost as time passes. But do we expect like them to be like more preserved if, I don't know, we dig deeper in the ground? Because I remember like there was a mission that attempted to do that, but it was a little bit of a fiasco because.
Definitely. So the problem with the surface is threefold. One is galactic cosmic rays, solar cosmic rays, and the perchlorates. There is a lot of things that can degrade organic matter. This is not really known, but I think that it is thought that the perchlorates, once you go deeper, they dilute. So there might be less oxidants. And also UV radiation, and in general, solar cosmic rays don't have the penetration capacity that galactic cosmic rays have. So if you go into the subsurface, you don't get rid, but you reduce two important factors. Now, galactic cosmic rays can penetrate up to 2 meters, and the Rosalind Franklin can drill up to 2 meters. So we're there. But it's true that the subsurface is definitely the place to look for. It's if you expect to find preservation, you need to dig. And if you find a matrix like salts or ices in the subsurface or phyllosilicates, even better.
Thank you, Pablo. Great talk.
Thank you.
I have another question. What is the oldest fatty acid sample found on Earth?
The oldest fatty acid sample found on Earth, I don't know. But here's the thing. So the oldest biological lipid found on Earth in Australia, it's not a fatty acid, actually. I don't remember do you remember beta carotene, the structure of beta carotene? It has 2 rings at the end, right? So they found a ring of one of these pigments in a formation that is 1.7 billion years old. Now, this is unequivocally biological. This is the oldest biological lipid for sure on Earth. There are fatty acids that have been found in samples that are older than that. So reaching the 2 billion year scale, but you cannot say this is the oldest biological lipid, because fatty acids have been detected in meteorites and in carbonaceous meteorites and chondrites, and that means that we're forced to ponder the possibility of abiotic generation of fatty acids. Even in the Earth's mantle, we know that hydrocarbons are generated by Fischer-Tropsch generation in the presence of oxidizing microenvironments. Abiotic fatty acids are possible. So we cannot say that these are the oldest. But to answer your question, I think it exceeds the 1.7 billion year old timescale.
Thank you.
Thanks.
Other question? Yes, Antonio.
Thank you for your talk, and it's very nice to see this overview, to have all things together. I just have a question because I don't know if I'm right, but I think Curiosity have an instrument inside as part of the SAM, the inner laboratory that it has, that is able to distinguish between carbon isotopes. But I don't know if the capabilities are limited and it's not enough to be able to assess on the origin of those carbons, but I don't know.
Yeah. I don't know much about this, but it has a capability of measuring carbon-13, bulk carbon-13. So a sample can be measured, and the whole sample has a general carbon content, and I think the Curiosity can measure the total carbon-13, the bulk carbon-13. So in fact, I think that the average measurement, there is an average value for the contents of carbon-13 in Martian regolith, but that's bulk carbon-13. If what is really good is the compound-specific isotopic analysis. So this analysis allows us to determine the carbon-13 content of each molecule, and that gives us a way better overview of how that carbon came to be, the metabolic pathways that gave rise to these components. So that I'm pretty sure the Curiosity cannot do, and that's why that would be, in my opinion, one of the most interesting analyses that you can do after Mars sample return. Yeah, that's a good question. Thank you.
Yeah.
Thanks, Pablo, for this fascinating talk.
Thank you.
I have a question. I'm not an expert at all in searching for life on out of the Earth. So I guess that if you're looking for lipids or whatever, it is because you're putting your eggs on the basket that says that perhaps detecting real life is going to be very hard. So you're looking for a life that disappeared in the past. And so regarding the state of the art, what the community thinks of and your own opinion, I would like you to tell me whether you think that it is, or the community thinks that between these three possibilities, which is the most probable, that there was never life on Mars, that there was life, but it disappeared completely, or that there was life and there should be something somewhere. What do you think about that?
I like it. I like the question a lot. And this is based on what I've read and my opinion, obviously. But okay, so just to be realistic, the possibility of existing life on Mars is not zero. There are refugia on the planet, like caves, and there are places that lava tubes, etc. That, you know, why not? We don't know anything about them. But I think that given what we know of Mars right now, I'm going to say that it's likely extinct. Now, whether there was life or not, if we look at Earth and we look at what we think Mars was, if we look at Earth, it seems that sometimes I think that life is not a miracle, that much of a miracle. Sometimes I think that with a given chemistry and with a given chemistry and a given incoming or interesting organics from meteorites, you can form at least interesting complex precursors to life and at least basic life. So I think that the possibility of a primitive life that did not evolve much and that was pretty simple, that was similar to prokaryotes, I think I'm going to say that's the option I choose from the three, and that life is extinct. And that's why we're looking for biomarkers of extinct life. But the good thing is that looking for biomarkers of extinct life, opens up the possibility to detect biomarkers of existing life. So you're still, you can cover that. But yeah.
I think it is quite hard that once that life has appeared, I think it's hard to make it disappear. But
Okay.
it's true that there has been so many years since then that perhaps you're right.
Mhm.
I don't know. Thanks a lot.
Yeah, good question. Thanks.
Are there any other questions here in situ? No? On Zoom? No? Okay, I have one more.
Okay.
So I wrote because I didn't remember all of the names. So when you show the silica sinter comparison
Yes.
between on plate and El Tatio Chile,
Yes.
it's just a visual comparison?
It's what, sorry?
Or there is a visual comparison, or there is also an analysis done on this? So it's just like
Yeah.
look into the pictures and they look similar.
Just
So
gonna go back to it. Oh, it's so far. Sorry. Yeah.
I just wanted to
Yeah.
know if there
Yeah.
was also an analysis.
No, I don't know. Honestly, I'm not really well aware of the instrumentation that Spirit carried. I know that it didn't carry anything. So it was not drilled. There was no laser shot at it. There was no sample taken from it. I know there was a photo. There was a photo taken, but if there were any other analysis, I am not sure. If anyone knows, I would love to know. But this is part of the reason why this site is an interesting site to return. I think there's interest to sample this
Because
silica.
I was wondering also if the visual similarity can be also done because of the weather. So if there is can be some environmental condition, and then at the end, you are lucky, and then they look similar, but maybe geologically they are different.
Absolutely. So the silica sinter in Chile is actually biomediated. The shape is actually because of the organisms living there. One has to assume that the shape of the silica sinter on Mars is weathered. It's caused by flowing water and wind. But the interesting thing is that the only interesting similarity is that they actually might have the same chemical composition, the minerals. And the mineral we know is good for preservation. So it might have been weathered and it have acquired that shape most likely because of Mars, of the abiotic factors on Mars, but maybe it has been able to retain some sort of interesting chemistry from the past.
Okay, thank you. I'm also very interested in the isotopic analysis, but
Yes, I remember.
yeah.
Still learning about
But we can
Yes.
talk offline maybe.
Sure.
Okay, if no other questions, so we can thank Pablo again for this very nice seminar. And thank you all for coming.